This video is to walk through how we do the process of transcription and translation on paper. So I have a DNA sequence here, a double-stranded DNA sequence, and in the process of transcription, we are going to copy one of the strands into RNA. And then in the process of translation, we are going to convert that RNA sequence into an amino acid sequence. On the right-hand side, I have a codon chart. You should have one of these in your book or in your assignment, and we will use that as sort of a code or a key for going from RNA to amino acids. When you transcribe a sequence of DNA, you always want to use the template strand. I'm just going to label one here. Usually you'll be given the template strand or you'll be asked to figure it out. But I'm going to tell you that we're just going to use the top strand here as our template strand. So we have our double-stranded DNA molecule, and we need to make a single-stranded RNA. Now remember that in RNA, we use uracil, not thymine. So that means that if we have an A in our DNA, we need to insert a uracil in our RNA. The opposite is not true. If we have a thymine in our DNA, we still use an adenine in our RNA because T always pairs with A, and we do put adenine in our RNA. So let's go through making our complementary strand. So if we have a T, we're going to insert an A. If we have an A, we're going to insert a U. And if we have a C, we're going to insert a G. C pairs with G, G pairs with C, A pairs with U, T with A, C with G, G with C, and so on. So everywhere I have a C in my DNA, I insert a G. If I have an A, I insert a U. If I have a G, I insert a C. And if I have a T, I insert an A. So now we're going to take this RNA sequence and we're going to convert it into a protein. So I'm just going to rewrite it here. Now notice I've used the RNA with the uracil. So if you get to this point and you don't have uracil, you need to double check that you've made your sequence correctly. And you need to double check that for your DNA, you insert a U instead of a T. All right, so let's use this codon chart. So on the left side, we have the first letter. So it's going to correspond to the first letter of our code. On the top, we have the second letter. On the right, we have the third letter. So I'm just going to look A, U, G. This is my first sequence. So first letter A, second letter U. So A, U, G. A, U, G. That's our first codon, and that corresponds with the methionine amino acid. You can just write the abbreviation. Now this methionine is actually a start codon. AUG is our start codon. If we insert a methionine in the middle anywhere, it's just a methionine. Next one is GCU. That corresponds with alanine. AGC, serine. UUG, leucine. And so on. I will just fill in the remaining ones. Now you'll notice that I put in 2 leucines here from 2 different sequences. Now, this is called redundancy. And leucine is actually coded by 6 different codons. You'll notice most of these are coded by 4 different ones, and there's some variation there. This allows for mutations to happen in the DNA without the amino acid changing. So it's sort of a way to reduce the effects of amino acids. So I'm going to do another example. I'm not going to narrate it too much, but I'll just walk through how it works. C codes for G,